This video is on some of the techniques that we're going to use to identify and diagnose some of the genetic disorders that we've discussed in this year's chapter on human genetics. So genetic screening is something that, as you can see here, it's just an examination of a person's genetic makeup. Many times you do genetic screenings prenatally, so parents would go in and have their child that is unborn figure out what the genes might be and or the chromosomes that they're possessing to kind of prepare in case there are any issues that might arise so they can be well prepared upon birth. Other times, if someone does have a suspected genetic condition, they can do it post-natally where they weren't aware of it and weren't suspecting it previous to the birth. So whether that happens at a baby, a teenage, as an adult, you can get a genetic screening at any point. And basically anything that they're now doing with 23andMe and all these other companies, they're doing genetic screening on you when you send your samples off to them. A genetic counseling position is something that I know in class that I do where you had to write the letter to a parent after finding out that their child potentially had some type of chromosomal abnormality or gene mutation. So it's just a form of medical guidance. You don't have to be a doctor. There are programs that maybe if you are interested in, you can see undergraduate and graduate programs to have this ability to basically guide people through their issues that might arise because of some type of genetic predisposition. In order to do that, what they're going to do is they're going to run a test. And the most common test that parents will get is an amniocentesis. This is mainly done for mothers that have maybe been pre-diagnosed to have a higher risk of having children that might have chromosomal issues, or any parent that used to have it at 35 years of age. They might have pushed that back in recent years to somewhere closer to 40, just because the risk of these chromosomal abnormalities increases with maternal age, and that all relates to meiosis as that reason. But what they'll do is, you can see there's a picture here of a pregnant woman, and they're going to take cells from the amniotic sac. So there is a sac that floats around that the baby is inside, and inside that cell or that sac there is the baby with fluid around it. So they do a little ultrasound to make sure that they're not going to puncture anything for the baby, and so they take this long needle. I personally obviously have never had this done nor seen it, but I can imagine that it is somewhat of a shock to the system to take this long needle and insert into the amniotic sac, remove the cells, and those cells would then be fetal derived, so you can take any one of those cells and analyze the nucleus. So this is done at 14 to 16 weeks of development. The karyotype is performed, and this is where you would see if there is any missing or extra chromosomes, and then the parents would have up until 40 weeks to decide what's happening, and they have plenty of time to prepare for any news that they might have been given. So amniocentesis is the first. The second is something called the chorionic villi sampling, also known as CVS. It's done a little earlier, but there are more risks involved. This is not a thing that every parent does, because there comes a time where you have risk analysis. The possibility of miscarriages and things like that can arise because of sticking needles and going into the sac where the baby is being stored. So it's not always the most safe thing. So that's why you only do it in dire needs or suspected issues that might have already been suspected from some type of doctor's visit prior. So the CVS, they take a sample of the tissue, so it's a little less invasive in terms of a needle. So they are going to go through the vaginal canal, and they're going to take what's called the chorion. And the chorion is also another layer of cells that is fetally derived, and they'll do the same thing. They're going to run a karyotype. So you can't tell if the baby's going to have blue eyes or brown eyes or something like this, but you'll tell if they have an extra chromosome such as trisomy 21, or they're going to have Turner syndrome or Klinefelter's, any one of those disorders that we call non-disjunction disorders. Non-disjunction disorders being those that have happened because chromosomes failed to separate during anaphase. So I've discussed this in videos that talk about mutations. So if you have any issues or are confused with that, you might want to refer to one of my mutation videos. And the final thing that most parents will get, because it's so not invasive to the child, is a ultrasound. And maybe you have your ultrasound. They're getting pretty interesting these days, very 3D. This is definitely not a newer ultrasound picture, but all you get from the ultrasound is an image of the fetus, and from there It might spark doctors to say we maybe want to do an amniocentesis, but you generally do this for parents that just want to know the sex of their child. And from there, you might be able to see severe abnormalities, and or multiple fetuses. Again, you're not going to determine what eye color they have. You won't determine if they have gene mutations such as cystic fibrosis or anything like that. So this is just a very preliminary test that you might do, and then some of the other follow-ups might be done because of the outcomes of an ultrasound. So that kind of is just a very simple how they most commonly are going to diagnose prenatally in the process of birth. Okay.